Hello, good morning, everyone. So today we are going to continue with our third plenary lecture. My name is Satirah, and I will be chairing for this session. So this morning we will have a talk from Professor Michael Fenech from University of South Australia. So allow me to introduce about Prof. Michael. Okay. So Prof. Michael Fenech is recognized internationally for his research in nutritional genomics and genetic toxicology. For 10 years, his lab has developed the lymphocyte CBNN, which is cytokinesis block micronucleus assay, which is now a standard method used internationally to measure DNA damage in human and mammalian cells. His method has been published in the Nature Protocols. His key goal is to determine the nutritional and environmental requirements for DNA damage prevention. His research is focused on the impact of nutrition and psychological stress on chromosomal and telomere integrity, personalized nutrition for dementia, prevention and cancer growth cell. He has a H index of 92, okay, based on 40,051 total career citations according to Google Scholar. Ladies and gentlemen, let us all welcome Prof. Michael Fenech. Prof?
Good morning, everyone. It's a real pleasure to be here with you this morning. I look forward to sharing this presentation with all of you. Thank you, Satirah, for the introduction, and to the Malaysian Society of Pharmacology and Physiology for inviting me to be here with you. Can you see my presentation?
Yes, Prof.
Okay, good. So the title of my presentation is Nutrigenomics from the Laboratory to Clinical Practice. The initial observation that we can see here from this slide is the great diversity in the foods that people eat. So these are actually photos taken by journalists of a family in a different country and the foods that they eat in a week. And what you can see is how great the difference is between the amounts of foods eaten, the kind of foods eaten, and the extent of the foods, whether they are processed or not. And we really don't know what the impact of these different dietary patterns and foods is, and how it impacts people from different ethnic and genetic backgrounds. So the field of nutrigenomics is aimed to try and understand the impact of different dietary choices, how that impacts health, and ultimately to use this knowledge to be able to optimize the dietary pattern to improve health, both at the population level and at the individual level. And it's important to state right from the beginning that even within a family, the genetic differences can be large enough so that the same dietary pattern can impact health differently within members of the same family. Now, the field of the concept of nutrigenomics is really to understand the impact of diet on the genome and metabolism. And diet can impact the DNA itself. It can cause alterations at the epigenetic level, which are really marks on DNA that can, or the histones in DNA, which can alter the expression of genes. This will then impact on the kinds of proteins of the RNA and the proteins that are produced within the cells, and ultimately impact the metabolites within the cells, which then affect how the cell functions. Furthermore, the metabolites that can be generated will depend on cofactors and transcription factors in the diet. And these can have a direct impact on the DNA in determining its stability or the expression of genes in the DNA. Now, when we consider health, we can, of course, look at it in many different ways. One can look at it from the point of view of prevention of disease, but one can look at it also in terms of, for example, healthy aging. And in fact, we do start aging right from the first day of life, because damage to DNA, for example, can make cells, even when you are still at the 1 or 2 or 16 cell stage, it can still make the cells defective and therefore age. Now, there are 9 hallmarks of aging. It's 10 if you include inflammation in the scheme here. But the most fundamental ones are the ones that affect the DNA. In other words, genomic instability, telomere attrition, epigenetic alterations. And this is because this affects the stem cells, the ability of the cells to regenerate themselves, and also to senesce. Now, there are a number of biomarkers for DNA damage. And what I'm showing you here on this slide are the biomarkers that are best established and best validated in terms of their association with disease, association with nutritional factors, and in predicting health outcomes. Now, the original method was to measure chromosome aberrations, which can be done using human lymphocytes. Another method, a much simpler method, is to look at micronuclei in cells. Micronuclei originate from fragments of chromosomes or whole chromosomes, as I shall explain. Quite a lot of work is done on telomere length, which is the TTA GGG repeat sequence at the ends of chromosomes, which is needed to maintain chromosome stability, or DNA strand breaks. Also, one can measure DNA adducts, such as oxidation of guanine in DNA or methylation, and also other groups study damage at the mitochondrial DNA level. The mitochondria have their own DNA, and one can study the extent of fragmentation in the mitochondria. It's impossible to talk about all of these techniques, so I will just focus on one, which is the one I've done most of my work with, which I also developed, which is the cytokinesis block micronucleus assay in human lymphocytes. So essentially what happens is when this is done with peripheral blood lymphocytes, when they are damaged, that damage will be converted to chromosome breaks or abnormal chromosomes like dicentric chromosomes. These, when the cell divides, they will either form separate fragments between the daughter nuclei, which lag behind and therefore form become their own membrane, forming a micronucleus, or there might be nucleoplasmic bridges from dicentric chromosomes, which you can see as a bridge here between the binucleated cells. To do this method, you have to block the cell at the binucleated stage, because that's at the point when these lesions in the chromosomes are expressed as micronuclei and or bridges. Most of the work that has been done to date has been with micronuclei, and in fact, it's turned out to be the most robust cytogenetic technique for these kinds of measurements. In fact, one of the very first observations made using the cytokinesis block micronucleus assay is that the micronucleus frequency increases with age. So when we are born, the frequency is very, very low, about 1 to 2 cells in 1,000 have a micronucleus. By the time in the teenage years and early 20s, the DNA damage rate has already increased quite a lot, about threefold or fourfold, depending on the individual, and keeps on increasing with age. And those who actually live longer appear to have a lower frequency of damage than would be predicted from the frequencies in the earlier age groups. And this is probably because the ones with the high levels of damage actually do not survive beyond age 75, for example. Now, we know that the risk of developmental and degenerative diseases and the rate of aging increases the more micronuclei and other types of damage that the cells have. So there is the question from a public health point of view of knowing what is the threshold of DNA damage we should allow in a public health sense, and can we design exposomes, this means environmental exposures, including nutrition, that enable us to stay below this threshold. In this context, given the COVID situation, one also needs to bear in mind that even viral infections can also damage the genome and contribute to this level of damage that we see in people. Now, from the nutrition point of view, it was already known back in 1971 that malnutrition can cause a large amount of chromosome damage. This study was actually published in Nature by Armendariz et al. And what they did was they studied the level of chromosome aberrations in the lymphocytes of children who had protein calorie malnutrition. So they were looking at this from the macronutrient point of view. And what they observed was that the frequency of chromosome aberrations in those that were deficient in protein intake, who had very low protein intake to the extent that they had a clinical phenotype of kwashiorkor or marasmus, increased the level of chromosome aberrations being deficient about 5.5-fold relative to well-nourished infants. And this increase in chromosome aberrations is as large as that induced by doses of radiation that would be unsafe. So it tells you already immediately that malnutrition can cause as much damage to the DNA, to the chromosomes, as environmental factors that we know are carcinogenic, like ionizing radiation. But it's not only undernutrition that can increase DNA damage. It can also be overnutrition. So for example, in this study here done in Italy, not that long ago, they studied the level of DNA damage in lymphocytes in obese and overweight children relative to normal weight children using the micronucleus assay, as explained before, but also using another biomarker known as the gamma H2AX assay that detects DNA strand breaks. And what you can see here is that the overweight and obese children had an 8-fold increase in gamma H2AX, in other words, in DNA strand breaks, and a 2.7-fold increase in micronuclei, again indicating that excessive caloric intake and poor diet can increase DNA damage dramatically. However, nutrition is not just about macronutrients, it's also about micronutrients, and in particular vitamins and minerals, which are required as cofactors in important DNA metabolism pathways. For example, folate, B12, vitamin B12, and choline are needed for maintenance of methylation of DNA, which is needed to maintain the structure of the chromosome, as well as for maintenance of gene expression control. Some vitamins play an important antioxidant role, either by the fact that they themselves are antioxidants, such as vitamin C, but also because they are needed as cofactors of antioxidant enzymes. And in this case, minerals are quite important, such as zinc and manganese. Other cofactors are other micronutrients are needed as cofactors of DNA synthesis and repair. These include magnesium, zinc again, and niacin. And these are obviously important because if your DNA repair is not, and DNA synthesis is not functioning well, this can lead to DNA breaks, unrepaired DNA adducts, which in turn, as well as telomere shortening, for example, and this in turn impacts the chromosome stability, leading to the telomere end fusions, anaphase bridge formation, chromosome breaks, micronucleus formation, and then leading to the chromosome instability phenotype and the aberrant karyotype. Now, there are many studies, of course, that have already been done about vitamins and minerals when they are deficient and how this impacts genome integrity. I'll just show you just some data from South Australia that we obtained some years ago using just dietary food frequency questionnaire data. And what you can see here is the impact of intake of foods, of vitamins and minerals from foods on the DNA damage level observed. And what you are seeing here is a comparison between the mid and highest tertile of intake relative to the lowest tertile of intake, the reference point being this green line, the horizontal line. So you can see here that increased intake of foods rich in vitamin E, calcium, folate, retinol, nicotinic acid is associated with a reduction in the DNA damage level by up to 45% in the case, for example, of calcium and nicotinic acid. With beta carotene, it's a U-shaped curve. You need beta carotene, probably mainly because of the antioxidant properties of it. In this case, protecting DNA damage, but too much can become pro-oxidant. And an excess of riboflavin, pantothenate, and biotin, this is foods rich in these vitamins, appears to aggravate the DNA damage level. And this is probably due to interactions between inadequate, for example, amounts of folate and excessive riboflavin can make the form of folate needed for DNA synthesis to be unavailable. And this can induce breaks. We are still trying to understand these interactions between nutrients, but we know that they have a strong impact as well as the interaction, nutrient-gene interaction. Now, having knowledge about which foods are rich in the nutrients required for DNA damage prevention is critical because it enables us to know which foods one should choose to maintain our DNA in its most stable state. In other words, to optimize genome integrity. So you can see quite clearly that almonds, for example, wheat bran and cheddar cheese and broccoli are rich in the nutrients we know are protective. But other foods such as beef and bananas and others may not be so as rich. So one has to, they might be rich in other factors that we did not study in this particular investigation, such as fiber is very important contribution from bananas. But this is just illustrates the point that having this knowledge can help us choose foods more carefully, and particularly those that are nutrient dense for those nutrients that are genome protective. Now, For the rest of my talk, I'm going to be speaking more about the personalization of nutritional requirements. So since more than a decade ago, there's a lot of interest generated about the genetic aspects of nutrition. In other words, can we, based on the genetic makeup of an individual, determine what's their optimal diet? So for example, if you had a defect in absorbing a certain vitamin, perhaps you need more of that vitamin to be able to get the right amount into the body is one of the most common examples. Now, in the case of nutrigenetics, we are here really looking at the same four aspects, but in a different way. So we require the genetic information to understand the predisposition of an individual. In other words, that they might be predisposed to be unable to absorb a particular nutrient or to utilize it, right? The other aspect is the epigenetic aspect. We know that when we are, through the cues we get from maternal nutrition during pregnancy and during early feeding and so on, and later in life as well, we acquire epigenetic marks that may switch on or turn off certain genes, and which may then affect the ability to utilize nutrients. So knowing the epigenetic side of things helps us to understand programming. And then one can use the transcriptomic and proteomic biomarkers to measure molecular response, and metabolomics and genomics to show efficacy and safety. So the DNA damage markers are also important to understand whether the foods that you are eating may cause harm or good to the genome. Now, the field of nutrigenetics is really not that new, because in fact, back in 1934, it was already established by Dr. Ivan Fowling that having a diet rich in phenylalanine can cause a very serious condition known as phenylketonuria. And this is due to a genetic mutation in phenylalanine hydroxylase, which blocks the capacity of cells to convert phenylalanine to tyrosine. As a consequence, phenylalanine then becomes converted to phenyl pyruvate, which is toxic by causing a defective energy, lipid, nucleic acid, protein, and protein metabolism. So defects in energy, lipid, nucleic acid, and protein metabolism are induced by phenyl pyruvate and other metabolites that are derived from it, like phenyl lactate and phenyl acetate. This gene, the phenylalanine hydroxylase mutation, causes this problem, and this particular gene is located on chromosome 12. Now, it soon became evident that that the way to correct this defect is to actually advise eating a diet that is low in protein and low in phenylalanine in particular. So a low protein, low phenylalanine diet is the solution to this particular defect. And progressively over many years, more and more genetic defects have been identified. There's tens and hundreds of them now that we know about. But the question is, how do we utilize this knowledge, and is it effective? So to illustrate this concept, here is a nice diagram by Corella and Ordovas, which this is a hypothetical situation where you've got a statistically significant genetic effect on the whole population. For example, for people who are, in this case, carriers of the A allele versus G carriers for a particular mutation in a gene. And when you observe this, these differences, you suspect that maybe there's a difference in people who carry the G allele versus the A allele, depending on the diet that they consume. And And in a typical situation, you would find that with a particular, with diet 1, there's no difference between the genotypes, but with diet 2, there is. And therefore, there is clearly an interaction, a diet gene interaction. Another example would be where you might not see a difference between those carrying the A or the G allele. But that doesn't mean there might not be a genetic difference, and that could happen if the effect goes in opposite directions for different diets. And in this manner, this is the only way you would find out that the genotype difference actually has an impact. And I'll just give one example here illustrating this second example. It's one done by Chao Chiang in a Puerto Rican health study in the United States. Essentially, what they observed was that DNA oxidation level, measured as 8-hydroxydeoxyguanosine in the urine, varied depending on the mutation in the MET1A gene, in such a way that those who were carrying the A allele were the ones who benefited from vitamin B6 in terms of prevention of DNA oxidation. And this was explained by MET1A being important to convert methionine to SAM, and from which homocysteine is derived after it donates its methyl group. And homocysteine conversion to cysteine and glutathione requires B6. So those who benefited were the ones who could actually generate more glutathione as a consequence of the activity of MET1A and B6 driving the homocysteine to cysteine reaction. Now, of course, although we have a lot of nutrigenetic information, not all nutrigenetic data can be applied or is relevant to use in practice. So the Nutrigenomics organization in Europe, in collaboration with other groups worldwide, we put together this framework of how, as a guideline of how to determine which polymorphisms in genes are actionable. So first of all, the study quality has got to be high. In other words, particularly an intervention would be high evidence that actually prescribing a particular dietary pattern generates a benefit for those of a particular genotype, that there's clearly a gene-diet interaction, and that the impact is actually direct, it's not an intermediate effect, that the nature of the genetic variant is such that it actually causes the metabolic defect, and it's biologically plausible, and that overall the scientific validity score for the gene and diet interaction for that particular polymorphism is high. Now, another aspect, now more and more, with more knowledge, it's become evident that actually the metabolic phenotype is probably even more important than the genetic background, and I'll show you some examples regarding this. So, for example, studies have been done where people were given a particular food, and you're looking at the changes in the metabolites after eating the food, and it's been observed that there's a great variation, for example, in the glucose, insulin, and lipid levels in the blood in the postprandial phase, that is during the 4 to 8 to 12 hours after eating. And the reasons for that was not known until recently. So the group by David Zevi et al. In Israel did a big study on 800 people who were fed standardized foods of different carbohydrate, lipid, and protein composition, and measured the microbiome, did blood tests of these metabolites in the blood, and dietary questionnaires. They measured whether they were, how tall and how obese they were, and also maintained the food diary. And using this data, they developed an algorithm that could predict the outcome of the metabolites after eating these foods, and for whom a particular food was hyperglycemic or not, for example. And in this way, they actually developed an algorithm that allows this to be well predicted and in fact is available commercially. However, the genetic aspects were kind of ignored in that study. And Bouke de Roos and Lorraine Brennan put out this nice paper on conceptually how we can determine which dietary patterns suit which person. For example, you could have a number of people being tested with 3 or 4 different dietary patterns. And from this, you can identify the responders and non-responders to the different dietary patterns. And from this, build a phenotype, metabotype, and genotype database that enables classification and proper matching of individuals with appropriate dietary pattern. And this concept and these concepts and ideas were taken a step further in this very important study, the PREDICT-1 study. The first paper was published actually last year. It was a study involving 1,000 people in the UK and 100 people in the US. And it was based on studying a baseline clinic visit where each participant had some food challenge tests done. They had genetic clinical assays done, blood spot tests, blood pressure and heart rate, anthropometry, and questionnaire lifestyle data. And then in the second phase of 2 to 14 days, they were given different test foods to eat at home on 2 occasions, and blood glucose, triglyceride, and insulin was measured. They also had a microbiome profile and so on. The outcome of all of this massive database was that well, first of all, the main outcome was that there was a large variation in the triglyceride and the glucose patterns after eating the test foods amongst individuals. And however, these changes could be explained by the factors that they measured. So it's interesting, for example, to know in the case of triglycerides, the most important predictive marker was the serum lipid markers that were measured at baseline, whilst genetics only explained maybe 1% of the variation, whilst the serum lipid markers measured explained about 20%. In the case of glucose, it was kind of different in the sense that genetic markers were the second best predictor of the response, explaining about 10%, whilst serum lipid markers had explained only about 4%, and so on. As you can see, essentially the main point is that for each biomarker that one could measure, there could be at least 10 different factors affecting it, and that the contribution of these factors varies for each biomarker. And furthermore, genetic variance only had a modest impact on the variability. They also could show, they could develop an algorithm from this data to predict what would happen in individuals, and they got a nice correlation here of the strongest one being 0.77 for glucose, postprandial glucose concentration, for example. And they concluded that prediction models using the most significant baseline characteristics can adequately predict postprandial glucose. However, they point out that the modest heritability of postprandial traits means that even in an unrealistically optimistic scenario in which most of this trait variance is explained by known DNA variants, it's unlikely that prediction algorithms using DNA variant data alone, which many direct-to-consumer nutrigenomics companies advocate, would succeed. So the bottom line is that the genetics alone would not be sufficient in a clinical practice sense, and that one has to use metabolomic markers together with the genetic data to really be able and other measurements to predict what might happen at the individual level. Nevertheless, the interest in personalization, personalized nutrition and precision nutrition has increased a lot. And many studies are going on to apply this knowledge to optimize pregnancy outcomes and the health in the first 1,000 days of the infant, to optimize immune function, which is particularly relevant today, these days, in terms of, for example, preventing the infection by SARS-CoV-2 and its consequences, metabolic disorders, in cardiovascular function, in neural health function, in liver and lung function, digestive and excretory functions, and cancer prevention. So thank you very much for listening to my presentation, and I'll be happy to answer any questions.
Okay, thank you very much, Prof. Michael, for the interesting talk. I forgot to mention earlier that Prof. Michael is currently an adjunct professor in Universiti Kebangsaan, Malaysia. So since we're running out of time, and I think if there's any question, okay, you can email the Secretariat, and we can pass the question to Prof. Michael. But I have only one question, for Prof. So how far and long are we into this personalized precision nutrition? What do you think, Prof?
Well, personalized nutrition, as I mentioned, is already being applied to certain conditions like phenylketonuria, right? So where there is really a strong genetic effect with a strong modification of phenotype. So this would be a highly penetrant gene, okay, and that the outcome is dependent maybe just on one enzyme in a pathway or two. In that case, then the impacts, if they are particularly severe, it becomes very important to be able to treat. And for some of those, there are strong recommendations on how you can treat that condition, depending on the mutation in that particular gene.
Right.
For more complex traits like, obesity, diabetes, and so on, where you have maybe 10 different genes involved and impacting, this becomes more complicated. And this is where you'll need to use metabolomics as well.
All right, thank you very much, Prof, for sharing your lifelong work, on nutrition genomics. Okay, so ladies and gentlemen, so we will continue our session, in 2 concurrent rooms. So we will have speakers, Prof. Renu Agarwal, which will be in this room. Okay, so for the attendees, if you're interested to listen to Prof. Renu's talk, so please just stay in this link. Okay, the recording might